Okay, next thing. Let's look at a few of the events of fertilization themselves. Okay, guys. Okay, you guys getting ready to split?
Just keep going.
Okay,
Yeah,
all right, cool. Okay. All right. Here's the egg. Here's our vitelline membrane. And here comes the sperm. Okay, the first thing, whether it's in the vitelline membrane or moving through the vitelline membrane, or whether things are in the egg jelly, it depends. Sea urchins, we have a set of receptors in the egg jelly. What's going to happen is you're going to get your first egg-sperm interaction. The egg receptors on the membrane of the sperm are going to bind to the appropriate sperm receptors that are either in the egg jelly or stretching through the vitelline membrane or what have you. So, okay, once those receptors interact, we are going to generate a signaling event in the sperm cell. Okay, we're going to have some interactions. One of the big things is we're going to fire off our phosphoninositide signaling pathway. And there also seems to be some type of protein kinase involved in this process as well. Okay. Now, several things are going to happen. Of course, when we have the phosphoninositide signaling pathway, we produce diacylglycerol and inositol triphosphate. Okay. Well, a couple things are going to happen here. Most of this is probably due to the diacylglycerol, which activates our protein kinase C. And that phosphorylates a whole slew of things inside the sperm. Now, protein kinase C is going to phosphorylate a number of things. It's going to cause a couple of things here. It's going to cause the opening of calcium ion facilitate diffusion channels. And it's also going to activate what we call a co-transport system. And meanwhile, our IP3 is going to cause, turns out the acrosome, aside from having enzymes, also has millimolar concentrations of calcium ions. Okay, so what's going on here? Signaling pathways activated. We get calcium released from the acrosome, flooding what little cytoplasm we see inside a sperm cell. We also open up calcium channels in the plasma membrane, allowing external calcium to get into the cell. And we're also going to activate a membrane transport, what we call a co-transport system of Na+ sodium ions and hydrogen ions. Now let's take a little look at this process here. Okay, now like all cells, you look puzzled, man.
What's the last word? Calcium release?
Calcium release from acrosome.
Okay.
Okay. So we're flooding the cells with calcium. We'll see what that calcium does in just a moment. But first, let's look at that co-transport system that gets activated here. That's a membrane protein. Co-transport is when you facilitate diffusion, which goes from high to low concentration, thereby is exergonic. You have facilitate diffusion of one molecule, and the energy released from that is used to do active transport of another molecule. So in a sense, it's a coupled reaction. Something goes from high to low concentration, and that powers the active transport of something different from low to high concentration. In this case, these sodium hydrogen ion co-transport systems are widely used. But what happens? Sodium ions rush in. Sodium ions are about 10-fold higher concentration outside than inside. It's even more extreme in a marine environment. So sodium ions rush on in, and the co-transporter pumps out hydrogen ions. Now, when you pump hydrogen ions out, you're getting rid of acid, so the internal pH goes up. So inside the sperm cell, the pH goes up, and generally we're talking it's starting out a little bit below neutral, maybe about like 6.8, 7 or so, and it'll go up to about 7.4, 7.5. That's maybe an 8-fold difference in H plus concentration. That increase in pH, we mentioned back in cell that some proteins are regulated by pH changes. So that increase in pH is going to lead to the regulation of certain proteins. We'll see what happens a little bit later. Now, meanwhile, we have calcium influx from the outside. Plus calcium released from the acrosome. We are flooding the cytoplasm with calcium ions. Calcium ions ordinarily in cell cytoplasm are 0.1 to 1 micromolar concentration. Now, to give you an idea how little that is, it would be like, the equivalent of taking 3 or 4 pieces of chalk and throwing it in an Olympic-sized swimming pool, and that's the calcium concentration you got. That's how low it is. So it's extremely low. Calcium concentrations outside are in the millimolar range, thousands of times greater. So we open up calcium channels, we release calcium from the acrosome, this calcium just floods the cytoplasm. Calcium regulates all kinds of things, and one of the big things calcium does is calcium ions, one of the many things calcium ions do, is they stimulate secretion of vesicles. In other words, the vesicles fuse with the plasma membrane and then dump their contents outside. And that's a common element of secretion of things. If you have allergies, and we're approaching allergy season, you can thank the secretion of histamine vesicles in the mast cells of your immune system for pouring out huge amounts of this histamine, this modified amino acid, into your bloodstream, giving you stuffed nose, itchiness, watery eyes, the whole 9 yards. Okay? You can thank calcium ions rushing into those mast cells for stimulating that secretion. Calcium ions are necessary for neurotransmitter release at the synapse. Calcium in general stimulates secretion. So we'll say vesicle secretion. And actually the reason that happens, if I just digress a little bit, okay, it turns out that vesicles are just beneath the plasma membrane. In one place or another, usually the vesicle membrane, you're going to have a protein. The one in neurotransmitter vesicles is called synapsin. It's found at the synapse. This protein is kind of unusual. It's an extended protein and can connect to receptors on the target membrane, like the plasma membrane. But what happens, in the absence of calcium ions, synapsin and its relatives, these relatives are found in the vast majority of vesicles. It's a very long, extended molecule. But you add calcium to this, it binds calcium, and then it folds up. It undergoes a massive conformation or structural change, and it folds up. It's kind of like stretching out a spring. Add calcium, the spring snaps back. And of course, what's going to happen is if this stuff is located on vesicles, and then all of a sudden you add some calcium and it shrinks like this, it's going to pull the vesicle really close to the target membrane. And now other proteins, especially in the target membrane, but in the other membrane as well, can merge together and cause the actual membranes to fuse. So calcium is necessary for almost all kinds of secretion. Whether it's neurotransmitter secretion, mast cells dumping histamine out, making you feel miserable, the processes of fertilization, or what have you. Okay, so, a question. Synapsin. S-Y-N-A-P-S-I-N. Plus relatives. Synapsin is the version they found in the neurotransmitter vesicles, but it's a family of proteins. It's related to a family of proteins that do the same thing.
Synapsin, that's the protein that's binding?
Yeah.
Okay.
Yeah. Okay, so, all right. So we have that.
Donors,
Oh, I'm sorry. Whoops. Okay.
Yeah.
Okay, I erased the signaling.
Wait, so the calcium adds it, causes the protein to, like,
To fold up,
Okay.
to compress up.
then what does that do to, like, how does that make the vesicle?
Oh, okay, well, let's picture this. Okay, I'll do a demonstration.
It's going to be good. Do you want me to put this on YouTube?
Okay, I do a crazy demonstration. Let's get it up in front of the whatchamacallit so we can actually see. All right, let's see, how about this? Okay, you are a plasma membrane. Actually, I'm curious. Let's see if I can shift this a little bit. Oh, I want to get a good view here.
Do you want me to hold it?
Well, let's see. Let's see if I can see. I'm trying to get past Jeremy's. Okay, hold it. All right. Okay, crazy demonstration again. Okay, well, what we have here is she is a plasma membrane. Okay, this is a vesicle membrane, and I am synapsin, or its relatives. So, I'm attached to the plasma membrane, to the membrane here. I can reach out and grab a receptor or two over here. And right now, I'm in my extended conformation. And this is getting painful. Now, somebody give me some calcium, quick.
Here's the
Okay,
milk.
thanks. Okay, now I come back myself and squish myself up, and you notice I'm pulling the two membranes together real closely, and we're going to have membrane fusion in just a second. Oh, we fused, yes, yes, yes. And my back will never forgive me for this stuff. Oh, man. Okay, thank you, thank you, thank you.
Thank you, Al.
All right, thanks a lot. Okay. All right, well, that gives a... Okay, that gives a basic idea of how does that help out? How this works? Okay, you can see I was pulling the two membranes together by this process, and we get close enough where the membranes can actually fuse. Did it help out, Jessica? Okay, cool. All right. I told you I'm not going to be hearing. If I was a suspicious person, I think somebody's trying to poison me. All right. Okay. So, we have a change in pH caused by the expulsion of hydrogen ions through our co-transport system. We have flooded the sperm cell with calcium ions, causing secretion. So calcium increase in the sperm cell causes the secretion of the acrosome. And we call this the acrosome reaction. So the acrosome, that giant secretory vesicle, fuses with the plasma membrane. I'm going to kind of diagram it here. Nucleus here, here's our flagellum. So the contents of the acrosome can rush out. What are those contents? The contents are enzymes for the most part. They include proteases, protein digesting enzymes, and glycosylases. Glycosylases are enzymes that attack polysaccharides. Guess what, folks? The vitelline membrane is made of what? It's made of proteins and polysaccharides. When the acrosome secretes its contents, those enzymes eat a hole through the jelly, if you got some. They eat a hole through the vitelline membrane, and now the sperm can follow in. So if we have So what's going to happen? We're going to eat a hole through the jelly. If you got some, eat a hole through the vitelline membrane, and the sperm will go right through that hole and come into contact with the actual egg itself. In a sense, anybody here ever been in the military? Okay, it's probably a good thing considering we tend to send them to places they don't belong. But one of the common things, a common kind of weapon used especially as an anti-tank missile contains what they call a shape charge warhead. And what it is you have a pile of explosives, then you have a copper cone at the tip of it. The thing detonates and it causes that copper cone to collapse and form a high-speed jet of liquid metal that travels at twice the speed of a rifle bullet, and that jet cuts through tank armor like a hot knife through butter. So something with just a couple pounds of explosives can actually penetrate through 2 feet or 3 feet of solid steel or several feet of reinforced concrete, even though it's not that actually large of an explosive charge. And then, of course, all the debris and stuff and hot gases go in and they kill everybody in the tank. Okay, well, in a sense, this is kind of like that. We release the acrosome and the contents, the enzymes, drill and blast a hole through the jelly, through the vitelline membrane, and now the sperm can follow in there. Now, the second thing that happens with the acrosome reaction is we mentioned there was a second set of receptors, of egg receptors here. These egg receptors are different than the original receptors that were on the surface of the sperm. The original sperm receptors on the sperm plasma membrane are very species specific. These are much less so. But these are the receptors that will actually bind to the egg plasma membrane itself. Now, people have actually done experiments where they strip away the vitelline membrane from egg of one species, artificially fire off the acrosome, and you can do that by just getting calcium into sperm cells. Fire off the acrosome in sperm of another species, put the two sperm, even though they are different species, together, and they will actually, in many cases, initiate fertilization. If you don't do that, and you put the stew sperm together, the first set of receptors won't recognize each other, and fertilization does not occur. So your species selectivity is done by the first set of the receptors. The second set is much less specific. Yes?
Okay, you probably said this, and I didn't understand. What is this thing that's coming off the back, like
Coming off the
line?
back? Here?
Yeah, what is
Oh,
that?
that's the flagella.
Okay, so
All
this
that
is
happened is you lost your acrosome. That's it.
Yeah,
Sperm
we
minus
just secreted
acrosome.
the acrosome contents out.
okay.
The other diagram's like totally separate.
Oh, okay.
Oh,
So I was trying to figure
sorry.
out how
Okay,
that
yeah,
went
this
in
is
with
the-
that. I was
So
like,
this is a detailed view, this is just a,
I thought that was the
back.
sperm, and then I was like, wait. Okay.
oh, okay, I'm sorry about
confused.
that. Okay,
Okay.
totally separate here. Okay, that was just
It looked
looking,
like it was going in backwards.
oh, okay.
That view.
All right, no, I won't do that. Okay, now there's one other thing that happens in invertebrate sperm, and that's triggered off. So in all these kinds of sperm, the acrosomal reaction is critical. That allows the sperm to penetrate through vitelline membrane, zona pellucida, same thing, or jelly or jelly, whatever. So it cuts a hole through all these outer layers, so now that sperm can go to the egg plasma membrane itself and initiate fertilization. But, invertebrate sperm has something else. And that's triggered off by our pH rise. Recall we had that internal pH rise caused by our sodium hydrogen ion co-transport system. Ugh. Okay, all right. Recall that many invertebrate sperm had a mass of actin behind the acrosome and from the nucleus. Okay, now that mass of actin that we find behind the acrosome, and once again, invertebrates only, but from the nucleus, contains either a bunch of twisted microfilaments. Microfilaments are also what we call efferfilamentous actin. In other words, the individual actin proteins forming this double twisted row like a set of rosary beads. Okay. And what keeps them twisted up is a protein that binds to it and forces it to twist. As a matter of fact, it was identified, and biochemists have great sense of humor, and they identify a protein, they get to name it, so they called it, because it twisted into a spiral pattern, they called it screwing. I'm sure they had a double entendre on that one. I remember laughing when I was at a conference where they first presented that stuff. But what it does is it holds this F-actin into this twisted coil, like a coiled up spring. The other alternative, you have individual subunits, we're called G-actin, and they're complex to proteins that prevent them from joining together to form an F-actin, a microfilament. At real high concentrations, especially the concentration you find here, those individual actin molecules should almost instantly join together to form microfilaments. But they have proteins that inhibit that process. And in fact, the protein has been identified as something called profilin. It binds the G-actin one-to-one and prevents it from joining together with other actins. And here again, biochemists with a sense of humor, they call the combination prophylactin. Prevents fertilization, at least in invertebrates, or helps prevent it. Okay, now, the thing with both of these proteins, the screw-in and the profilin, is they do not bind at high pH. You raise the pH, they fall off. Guess what we just did? We raise up the pH, the proteins fall off the F-actin microfilaments or the G-actin. And what's going to happen in this case? If you have these coiled up F-actin filaments, you lose that protein, they uncoil rapidly, so all of a sudden it's like taking a tightly coiled spring and all of a sudden stretching it way, way out. So all of a sudden those filaments increase many, many fold in length, often tens of micrometers, like 5 or 10 or 20 times the length of the sperm head. In seconds, I've seen this under a microscope. It's incredible. You see this, and all of a sudden this thing just grows out in a matter of seconds to something that's like 10 times the length of the entire sperm head. Now, the case with the G-actin, remove the profilin, and all that actin spontaneously forms new microfilaments in a matter of seconds. Now, what happens in this? Well, here you have your sperm egg. You fired off the acrosome. You have this mass of actin here. All of a sudden, it either springs into shape or polymerizes. It rapidly grows outward, and it pushes the plasma membrane containing our second set of receptors outward. So what happens is you get a cell extension like Pinocchio's nose that grows to tremendous length in a matter of seconds. And what that means, since this has the actual egg plasma membrane receptors, is it's going to interact with the plasma membrane of the egg a lot quicker than it otherwise would. So in this case, rather than waiting for the entire sperm to go all the way to the egg plasma membrane, you fire out that long extension. The receptors are going to connect and fire off fertilization before the rest of the sperm gets there. That's also an advantage if you're doing aquatic fertilization where there's a whole bunch of competing sperm. That helps you get there first. And we call this extension the acrosomal process. And I'll get to you in a sec. And once again, vertebrates do not have that. They don't have quite the level of competition. Jeremy, what's up?
The long thing, what makes it again?
Okay, once again, that's either coiled up microfilaments suddenly and catastrophically uncoiling. Okay. So if they're coiled up like a spring, imagine what would happen if you take a slinky. You know how much you can compact a slinky, right? Now imagine if you stretch it all the way out. That might be 10, 15 feet long, even though coiled up it's only about this long. Or the other thing is taking a bunch of single subunit actin molecules, G-actin, and all of a sudden joining them together in a matter of seconds and making these long microfilaments. And that pushes the plasma membrane out, and that structure is what we call the acrosomal process. It carries a subset of the receptors to the egg plasma membrane a lot faster. So it's a nice way of beating the competition here. Vertebrates do not have that. They don't have as high concentration of sperm, things like that, and internal fertilization also helps. So we don't really need that kind of stuff. But invertebrates, especially ones that do aquatic fertilization, often have it, where you're talking about millions of sperm and tens of thousands of eggs, and everybody a big free-for-all out there. Okay? So that's going to be our start of fertilization. We will finish the events, see the egg response of fertilization and egg activation. We'll finish that when we come back on Tuesday. And I will see you folks later.